Hi, I'm Nakissa Alayari from the laboratory of Rolf Bodmer in the Development and Aging program at the Burnham Institute for Medical Research.
I'm Georg Vogler, also from the Bodmer lab. Today we're going to show you procedures for the fluorescent labeling of larval and adult Drosophila heart structures.
We use this procedure in the laboratory to prepare the cardiac tubes for morphological and structural analysis via confocal or fluorescent microscopy.
In order to fluorescently label heart structures in adult Drosophila, first make a single cut between the thorax and abdomen in a semi-intact adult to separate body segments. Then, trim the overhanging ventral edges of the adult cuticle. Next, transfer and label adult or dissected larval specimens with primary and secondary antibodies. Following labeling, mount washed adult specimens by holding the cuticle edges and placing them heart-side down onto a drop of VectaShield on a cover slip. Invert the cover slip onto a drop of medium, flanked by 2 cover slips on a slide, to form a bridge, with the hearts now suspended upright. For mounting larval hearts, transfer specimens onto a slide with the ventral sides facing down. Mount 2 cover slips on either side of the slide. Finally, form a bridge with a 3rd cover slip by placing it first on the cover slip near the posterior ends of the larvae. The hearts are now ready for imaging. Before starting this procedure, prepare the following: ADH relaxing buffer containing ADH as described in